Hi, I'm Niharika.
And I'm Noya, and we are interns contributing to the COVID-19 Biomarker Knowledge-base, which is a collection of various biomarkers related to COVID-19 that are discussed in various scientific research articles. The collection is available freely on the OncoMX website. OncoMX is a cancer biomarker knowledge base. This project leverages the OncoMX data model to capture COVID-19 biomarkers. The format uses existing or widely accepted standards and ontologies, allowing for efficient extrapolation and extension of data to be used by other knowledge bases as well as clinicians and researchers.
A biomarker is defined as an indicator of normal biological processes, pathogenic processes, or a response to an exposure or intervention, including therapeutic interventions. Biomarkers have a wide range of applications depending on their usage in different stages of the disease. In the face of the COVID-19 pandemic, clinicians and researchers are exploring the roles of various biomarkers in the disease. They are classified as diagnostic, response, prognostic, safety, risk, predictive, and monitoring biomarkers. Now, moving into the process. Since the biomarker knowledge-base development was a crowdsourced project, volunteers were organized into reviewers and curators. Curators read publications and filled in tables. Reviewers then checked the entries for accuracy and moved them to the reviewed biomarker table. Curators read scientific journal articles and preprints published from January 2020 and synthesized information into a standardized format. The data collected was populated with ancillary data to harmonize the data entry with structured ontologies. Each curator would upload their file into a shared drive once every week. Reviewers would then compile the data from all the curators into a single cohesive dataset file marked unreviewed. Each entry was reviewed by at least 2 qualified curators who had experience in biocuration. Resulting entries were compiled into a reviewed file and ultimately published into the OncoMX website for public use.
The collection and compilation of biomarker data into the OncoMX knowledge base took place in 6-week cycles. Cycle 1 involved biocuration and cross-validation as well as the exploration of different combinations of biomarkers with best biomarker type or function in the disease. Interns were also organized into groups to work on the exploration of specific biomarkers that were not represented in the data list more than once or twice. Interns were also instructed to explain the work they had done in their groups each week in a small speech. This allowed for each group to gain a wider scope of knowledge in relation to more unique biomarkers. Cycle 2 involved biocuration and collection with the focus on documentation of new biomarkers to support a wider range of biomarkers published in the OncoMX database. There was also a concentration in collecting COVID-19 biomarkers overlapping with cancer, which provided information about clinical outcome or disease progression in patients with comorbidities. In addition, interns were organized into groups that carried out specialized tasks. Some were data collection, data visualization, data curation or review, and outreach. A newsletter and other representations of the project through various media were worked on by interns. Cycle 3 is focusing on similar subjects as Cycle 2 with interns divided into groups, each focusing on a different section of COVID-19 biomarkers. These include overlap of COVID-19 with cancer, overlap with diabetes, and new biomarkers. Each cycle also had weekly social sessions in which team exercises as well as talks by the Hive lab members took place.
We are trying to help researchers who are working on developing diagnostics or drugs by organizing biomarker data from publications into a standardized table and in a knowledge base. This is especially important in combating the issues of limited medical supplies such as ventilators because an evaluation of disease severity can identify which individuals can recover at home and which individuals need advanced therapies. Our COVID-19 biomarker resource is licensed by the CC-BY 4.0, which means it is open to any individual, facility, business, clinic, or anyone else for use. Some of the main goals and impacts of this resource will be for clinicians and researchers. This is a COVID-19 biomarker crowdsourced project where anyone with a biology background can participate. Anyone can join the project at any time. We are always looking for more people to help out. In addition, your hours are extremely flexible and will easily fit with your schedule. Currently, we are moving into Cycle 3, which will focus on the overlap of COVID-19 and diabetes for the research section. In addition, we will continue to expand the marketing aspect, which includes website development, newsletter writing, and video development. We will also delve deeper into data visualization. Email any of the following people and ask about the COVID-19 Knowledge-base project. This project is successful with the contributions of high school, undergrad, and graduate student interns, along with the Hive lab members.